So welcome. Today we're talking with Ian Gibbons, who nearly 50 years ago discovered the molecular motor protein dynein. So dynein is the force-generating ATPase that causes cilia and flagella to beat. It's also found in the cytoplasm of most eukaryotic cells, where it transports all kinds of different cargo, such as small membrane vesicles, the Golgi apparatus, the nucleus. It transports mRNAs and protein complexes. And in mitosis, it also plays an important role in constructing the mitotic spindle. So it's a very important molecule in cell biology. And well, I've known Ian for many years. We've collaborated on several occasions. But I've never had a chance really to sit down and talk to you about how this whole story came about, how you went about discovering dynein. So, well, let's go back to when it all started, 1963, and maybe you can tell me about what captivated your interest in studying cilia and flagella in the first place.
I was working as an electron microscopist at Harvard in the biology labs, and I was collaborating on a project involving looking at flagella in the various flagellated protozoa. And one of the days we happened to take some pictures by accident that showed a set of cross sections of cilia and flagella and the transition between the basal body or centriole up into the shaft of the actinine. And there were an enormous variety of fibrillar structures there that had never been described previously. And I felt that they must be doing something important. I knew that cilia and flagella were motile organisms. My supervisor at Cambridge, where I was getting my PhD, was John Bradfield, and he had actually written a paper on cilia and flagella mechanisms at that time. But then he left the field completely and went into finance. So I decided that when I saw these beautiful structures, I really wanted to know more about how they might work. There was so much structure there; it was far from obvious as to what it might do, how the different bits might interact.
So which of these structures did you think might be the force-generating entity, and then how did you actually go about figuring that out?
Well, I didn't get there all in one go. It took several stages. And one of the things was that after I'd spent a couple of years doing electron microscopy, I realized that one only got a rather limited distance looking at structure alone, because there was almost nothing known about the chemistry and not a great deal about the detailed motility. And so I decided to take on the biochemistry, try and go for it myself, though I had no experience in biochemistry. But I did have my wife, who was an experienced protein biochemist, working at John Edsall's laboratory at that time. And she certainly provided me with lots of help in getting started and knowing the important questions that I should be considering. So once I did that, then I decided that the Tetrahymena cilia were an interesting organism from which to isolate the cilia, since one can grow them up by the pound and isolate them. Actually, I bought myself a cream separator to harvest them, and we did get maybe about a pound of Tetrahymena in each culture that we were using. So then, from that, I used the method for isolating the cilia that had been developed by Sir John Randall's lab in London, and used that to isolate the cilia. His people had tried to look at the proteins, but they hadn't gotten very far, because they found that the proteins appeared to be almost completely insoluble. I suspected that the problem had been that the isolated cilia, after they fractured from the cell, the ciliary membrane healed over the isolated tip, and so the cilium was completely enveloped by the membrane, and so the proteins couldn't get out. When I got these preparations of isolated cilia, then I decided that the first thing to do was to get rid of the membrane, so I applied a mild detergent, digitonin. That did a wonderful job of taking off the membranes, and also showed that the structure remained intact. All the internal bits of the ciliary apparatus remained intact. And very importantly also, the cilia retained their axonal ATPase activity. Very little disappeared with the membrane. So we had the denuded axonemes. When I just put them into a concentrated solution of salt, the protein all completely dissolved. That showed that they were solid, that you had access to all the protein, but it wasn't what I wanted. I wanted to get some fractionation, and that I achieved by extracting them at low ionic strength with EDTA to completely remove any divalent cations. And when I did that, I found I got a very interesting separation of the protein into two fractions, a soluble fraction and the residual pellet. And comparing those two structures in the electron microscope, I saw that the extraction with low salt EDTA had removed all the central apparatus, the spokes, the two central tubules, and in addition had removed the arms, the inner and outer arms that ran along the lengths of the doublet microtubules. When I looked at the solubility of protein and ATPase, I found that that this had indeed achieved a good separation, because the solute, that soluble fraction, contained about 80% of the total initial ATPase activity, but only had about 30% of the total protein. So I'd obtained something like a threefold purification just by doing this simple extraction. And so since I had most of the axonemal ATPase present in the soluble fraction, obviously the thing to do was to look at it. And the only technique one had at the time, this was before gel electrophoresis, the only technique available was centrifugation. Started with an analytical centrifuge, and there were three peaks. One slow one at 4S, one at 14S, and one at 30S. When I ran the same material on a sucrose density gradient and assayed for ATPase activity, then I found that the ATPase activity was completely confined to the 14S and 30S fractions. The 4S had essentially none, though it was the largest fraction in terms of total protein. So then the question was, which of the various structures that had disappeared had the axonemal ATPase actually come from? And to find that out, I tried recombining. I took the soluble fraction, the pellet fraction, mixed them together in the presence of magnesium. And fortunately, I found that a major fraction of the ATPase activity that had been extracted in solution became rebound to the pellet fraction. Looking in the electron microscope, this showed that the structures that had reappeared when the ATPase activity became rebound were the two arms that stick out from each of the doublet tubules. So this was pretty strong evidence that the axonemal ATPase was indeed localized in these arms, the inner and outer arms on the doublet tubules.
So now you had this ATPase. You could show that you can add it back and it could reconstitute this really interesting structure between the two outer doublets. Next step, how did you come up with the name of this interesting ATPase?
Once we had the protein more purified, gave it the name Dynein. Dynein, but the dyne was a unit of force, the only unit of force present when I went through school. Actually, Barbara was the one who suggested the name Dynein. She suggested it one evening at home.
So at that time, how did you think it worked?
Well, there were two theories running. There were two possible mechanisms. One would be a mechanism involving localized contractions, that somehow the localized sections along the length of the microtubule might change the crystallized packing arrangement of subunits in a way similar to that which the bacterial flagellum does when it changes its shape from a left-handed helix to a right-handed helix under the influence of a chemotactic attractant. That would be one possible mechanism. Second possible mechanism was that the microtubules would slide relative to one another, and that this, when opposed by some resistance to bending, would cause the formation of a new bend. Difficulty with that mechanism was that to have bends of opposite directions at different positions along the length of the axoneme meant that the individual microtubules run straight along the length of the axoneme, but it meant that sliding has to be in opposite directions in order to generate bends of opposite directions at different positions, even where you're only thinking of a single microtubule. That seemed to be a major obstacle. And but I didn't take it any further. At that time, actually, looked in the text of the paper, I was actually leaning toward a contractile hypothesis.
So then what did you do next? It was time to do an experiment. So what experiment discerned between these two mechanisms?
After we'd taken the formality of publishing the more purified protein and the name, which is in 1965, then I realized that we needed to get more into the motility. And the Tetrahymena cilia were extremely inconvenient for doing that, because they're only 5 micrometers long, and although you can see them, you can see they're waggling vaguely when you look with a high-powered light microscope lens, but you can never measure accurately the parameters of waveforms. So that was when I decided that we should shift to sea urchin sperm flagella, because these are about 40 micrometers long, about 8 times as long as the Tetrahymena cilia. Their motility had already been studied by Sir James Gray, who'd shown that they propagate waves along the length of the flagellum. But we didn't have a real good source of them in Cambridge, Massachusetts, where I was then. And so we waited, and an opportunity came up for me to move to Hawaii, University of Hawaii, and decided that would be an opportunity to switch completely to using sea urchin sperm, since the sea urchins in Hawaii were sexually ripe all year round. You would have material for working with them year round without the complications that happen if you tried to do something like this in California or on the East Coast. So we moved to Hawaii. And then we started studying the motility in more detail. We first measured the parameters of the bending waves of the live sperm. Then we took the membranes off the way we had been doing for the Tetrahymena cilia, showed that they could be reactivated with ATP and propagated bending waves very nicely. Then the question was, what was the mechanical force actually going on inside the axoneme? And Keith Summers, who had my graduate student at the time, had the idea of using trypsin to weaken the structure. He thought that might do something. I had earlier observed that with the Tetrahymena cilia, they become swollen when you gave them ATP. They didn't slide, but they became swollen. So he tried to make this happen also with the sea urchin sperm flagella, but was completely unsuccessful. They didn't do the same trick. So he tried treating them with a bit of trypsin to see if that would get them to do the same trick. And well, it did, but it also did something much more dramatic, because they just seemed to disappear completely. If you took the demembranated isolated axonemes, treated them weakly with trypsin, and then diffused in ATP slowly from the edge of the microscope slide, as the front of ATP reached the axonemes, they just seemed to fade and disappear. So that something quite dramatic was going on, something that had not happened with the Tetrahymena cilia.
And you wanted to look at that directly and observe it.
I decided to use dark field microscopy to watch the disappearing of these flagella axonemes. So Barbara and I went back one evening, and we looked and we saw it, and we were amazed that it really slid apart. It was very dramatic, and that was really one of the high points, because it was obviously demonstrating a very fundamental mechanism in the interaction of a motor protein with the microtubule for the first time. And it was happening under our own eyes. One of the things we did was to get more evidence that the dynein was actually the motor driving the motility. The way we did that was by partially extracting the dynein arms. When we did that, we found that the beat frequency decreased proportionate to the fraction of dynein arms that had been removed. And then we could add this solubilized dynein back again, and the flagella beat frequency increased back almost to its original level as the arms became rebound. That was really the confirming evidence, the last nail in the coffin, or whatever you want to call it.
So we've covered the isolation discovery of the ATPase, dynein, to the in vitro demonstration that Dynein can slide microtubules, and that was the underlying principal mechanism that gave rise to cilia and flagellar beating. So maybe you can give us a quick tour of Dynein after that.
So at that point we wanted to characterize it biochemically, which we did, and by that time Lezioni gel electrophoresis had been discovered and developed, so we were able to start looking at the polypeptides that were present in our preparations, something that had not been possible when we did the original extraction of the Dynein, because of no gels available that were appropriate at that time. So that was one of the things. We worked up, we purified this ATPase, we started looking at its polypeptides. We eventually cloned and sequenced one of the polypeptides from the outer arm. And then after we cloned and sequenced it, we started looking at the different forms of it, because it turned out, as most people discovered with gel electrophoresis, preparations were always more complicated than you'd like to think. There are a lot more proteins present than one really liked to think. So we discovered that there were about 8 or 9 bands in the electrophoretic position of the Dynein heavy chains. And so we isolated different ones of them, studied how many there were, and that led to realizing that the Dynein family of the heavy chains alone consists of at least 9 subfamilies. And 2 of these correspond to the 2 cytoplasmic dyneins. The other 7 are axonemal proteins, and sometimes there's more than one representative of a family present at one time. So usual flagella axonemes like we were using contained 13 actually gene products. So we cloned and sequenced one of these genes, the heavy chain, the beta heavy chain of outer arm dynein, cloned little bits of others. But of course, as time went by, other people joined in, and so by now there's a real elaborate tree showing the 9 subfamilies of the Dynein heavy chain form independent subfamilies that were present even in the last common ancestor of all eukaryotes. In 1997, we wanted to retire from the University of Hawaii, worked less than full time. So we moved to California, where our family, to be closer to our family, our children, and there I started working on the cytoplasmic microtubule binding domain of cytoplasmic dynein. And by taking just that separate small piece that constitutes the microtubule binding domain and its coil stalk, which is only about 5% of the total heavy chain of the mass of the whole Dynein heavy chain, we were able to express it in bacteria, study its properties. We were able to show that by changing the register between the 2 strands of the coil stalk, we could modulate the affinity constant with which this expressed microtubule binding domain would bind to native cytoplasmic microtubules. Having got there and having this expressed a functional cytoplasmic microtubule binding domain, the next step obviously was to go on and crystallize it and to get a structure. Took a year, but Joan Garbarino in my lab did a wonderful job, and then helped especially with the crystallography. We joined Andrew Carter from your own lab, and together we managed to get a reasonable atomic level structure for the microtubule binding domain.
Are there any particular thoughts that you have, just looking back over this?
My general modus operandi, I think, has been to work with a technique for a while, and if it seems to be sticking, try climbing another side of the mountain. Change your technique and start afresh and approach it in a completely different way. I mean, and I think that's useful. I've certainly found it interesting. I don't know how well it will work today, where there are many more people in science and most problems are more popular. Competition might make it difficult for you to do that, but certainly it worked very well for me, and so I would encourage anyone else to try doing it if they have the opportunity. But the main thing is to have fun, enjoy what you're doing, and then with any kind of decent luck, you'll get a great result.